Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Tim Kelly from Oxford Biomedica Solutions.
Thanks, Leah. Hi, everyone. Good morning, good afternoon, depending on where you are. So today we're just going to talk a little bit about product quality for AAV vectors, which is near and dear to my heart, because that's what I've been doing for quite some time. Specifically, I think now it's probably clear, and most will agree, that product quality is really one of the major concerns with AAV and with gene therapy. And we all know there's been a lot of clinical holds, a lot of them due to CMC issues, also due to patient safety issues. So product quality is becoming really important. At the same time, regulatory hurdles are also rapidly increasing. This I think is also becoming apparent, and some specifics that are in high demand is really full breadth of analytical capability, qualifying QC methods, even validating methods in early clinical development. Having actual product quality attribute understanding for your product is also really important, but also valuable. And I think what we're also going to see is a push for more specifications being expected by regulatory agencies, and tighter specifications, of course. We all know it only goes in one direction, so we have to be ready for that. Third is demand still is very high. So lots of vector is needed, and it's still expensive to manufacture and to supply good quality vector to patients. So those are the three focus really areas that I'm going to try to address a little bit very briefly here. In terms of quality, and the way we look at it at Oxford Biomedica Solutions is have to have quality, but you also have to have the appropriate output. So supply, right? If you get a really teeny tiny amount and it's super high quality, that doesn't really do you any good. So you have to have both supply and good quality. To do that, I think there are really three keys. You have to have the right expertise, you have to have experience, and a wide breadth of capabilities. It's not enough just to have a great upstream process if you can't purify, if you can't get it filled appropriately and delivered to the clinic. You have to have the full breadth of capability, and you have to leverage that expertise, right? And that's something I think that's really important is working with a team and with someone who has all of that capability and has been through this process on different construct designs multiple times. There's also a lot of different product quality challenges, technical challenges, as I'm sure everyone listening has experienced. And it's, again, not enough to be able to address one of those issues. You have to really be able to address things from end to end, which is, again, why having a full breadth of capability is so important. One of the critical quality attributes that I think everyone focuses on now is empties. How many empties, how many fulls, which is really important and valuable. We've invested a lot of time into that, into our platform purification process. But that's only one, right? You have to have that control, but you also have to have a full method to cover all of the quality attributes. Not one is not going to be enough. I think it's very clear. The other one that's also getting a lot of attention now is potency, right? And I think appropriately. So if I had to call out the two, it would be empties or fulls. and potency as the most critical. But again, you have to have that full breadth of capability. At Oxford Biomedical Solutions, we have been developing a platform capability. It is a plug-and-play platform capability that we have run hundreds and hundreds of times now. We have a full breadth of capability in one single plant here in Bedford, Massachusetts. We have a team of 135 folks now who do our end-to-end capability from construct design all the way through to clinical supply. We have successfully executed 5 INDs. We've run our GMP scale, which is a 500-liter scale, now successfully 45 times since 2019. And as I mentioned, we run a 500-liter scale. It's a serum-free suspension process and a 2-column chromatography purification step. We have scaled that platform up to 2,000 liters and have run multiple constructs at that scale. And we do find that it is linear, so we're able to achieve the same titers and, as important, the same product quality at that scale. So how do we address these issues? And I would say, look, it takes a full team. It takes an army to address all of the issues. But the one thing that's important is really focusing on innovation and driving that innovation. And that's something we've been here for now beyond 5 years. We have 4 patent families covering upstream, downstream, and also formulation. To support that, as I mentioned, we have scaled this platform now up to 2,000 liters. We've run the platform more than 800 times, and at GMP scale, 45 successful batches have been produced. So that experience and breadth of capability across the whole team is something that's really important to leverage. The second piece is analytical methods. So again, having the full breadth, we have 43 methods that we've developed. We use a number of those for a routine GMP release, and then about 20 of those are actually for characterization, to understand what's happening with your vector as you're making it, which is really important. That's the only way to really learn, and learning for us goes hand in hand with innovation. We have a team, 135 of us now that are active, doing all of our construct design, process development, scale up, and GMP supply, and we've delivered 5 successful INDs. This slide just gives you a perspective of all those methods. I'm sure a lot of these methods, many of you have seen. What's important, I think, is having all of these methods and being able to qualify them for your unique construct. We've done that multiple times, obviously, to be successful with INDs, but we've also taken one of our constructs and done full deep characterization on the vector profile, which would include deep sequencing and aligning that with full potency assay as well, which again is going to be something that the regulators are going to be expecting, certainly at a pivotal scale, but maybe earlier. We'll have to see. So having this full breadth of capability is really important. In terms of addressing both product quality and titer or output yield, if you will, so what we've been able to demonstrate now is up to about 90% fully intact vector, and that, again, that's using the platform, suspension, transfection, and 2-column chromatography purification process. Orthogonal potency. So for phase 1, we have 2 potency methods that we use, and then we would expect before you go to pivotal, you will absolutely need a full potency method. We have developed one already. We're in the process of developing multiple other full potency methods as well. And then that full panel. You have to have the full panel now, so that breadth of capability is really important to get through regulatory agencies. We also have a 2 to 8 degree storage condition for on drug product with a proprietary formulation that we've developed. And again, that all of this can be available. We're offering our services to anyone who needs this capability. In terms of platform titer, so we're now achieving in the low E15 VIGs per liter. This is titer right out of the bioreactor. We've scaled that again to the 2,000 liter scale, so we have achieved E15 titer at the 2,000 liter scale as well. What that gets you is about 2E17 vector genomes of drug substance per 500 liter batch, which is a fair amount, and we think also very scalable to get to larger indications that will be needed for to grow this space in gene therapy much further and to address commercial needs. This slide just shows you kind of the progression, which I think is really important. So when we started doing GMP manufacturing back in 2018, you can see how much vector we were making, and then the progress, and then as you get to today, where we're at, which is the E15 titer, and that gets you to the 2E17 VGs per 500 liter equivalent. A lot has gone into that, a lot of innovation. That is also uses a dual transfection plasmid system that we've developed, and that also we've compared that to our triple transfection system. We offer both as a service, but we obviously are certainly pushing and leaning towards the dual because it performs so much better, as you can see here. So I think, again, being able to address supply, quantity, but also at the right product quality levels, really important, and important to balance all of those things to be successful as you move forward into the clinic. And so thank you very much for listening in, and I'm happy to also take any questions you might have.
Okay, great. Thanks, Tim. So before we move into the audience Q&A, we do have some questions for the audience. On your screen, you're going to see a couple questions. If you can go ahead and use your mouse to select the answer that best works. What is your expectation for percent full in your AAV vector? 100%, greater than 70%, 50%, or greater than 30%? And what scale do you expect to be at to meet your supply needs? 50 liters, 200 liters, 500 liters, or 2,000 liters? So again, if you can just go ahead and answer those questions on your screen, that would be great. And then we'll move on to your questions, which you can go ahead and enter at any time. So the first question is the purity the same for dual plasmid?
Yeah, so we've been developing the dual plasmid system for several years. We initially compared it head to head with the triple transfection system, and we've actually now done analytical comparability on 7 different constructs, 3 different serotypes as well, capsid serotypes. And we've taken one construct all the way through to in vivo animal testing to show that they were comparable, and in fact they're extremely comparable in all our analytics.
It looks like the next question is, can you explain how you were able to scale up your transfection to 2,000 liters?
Yeah, so I would say there's probably 2 components to that. One is having the bioreactor and transfection process, which is very tricky, but that's an area that we've focused a lot on and spent a lot of energy understanding that, characterizing it, and optimizing it so that it is linearly scalable, which we've been able to demonstrate multiple times. The other piece of that is having a team that is just well experienced with single-use bioreactors, and we're very fortunate. A large amount of my team here come with a ton of single-use bioreactor experience, including being the first to to actually commercialize and get FDA and EMA approval for a 2,000-liter single-use bioreactor for a protein. So marrying those two things together was really the key to allowing us to drive that and be successful.
Okay, and as a reminder, you can type in your questions at any time. And if we don't get to all the questions today, we'll be passing any other questions on to Tim, and he can follow up with you directly. So the next question is, how variable is the performance of your platform?
Yeah, so I mean that's a fairly broad question. What I would say is we don't see variability from batch to batch or even between scales, and we operate between 2-liter and 2,000-liter. So really, when we're operating, it's the variability of methods, the assay variability that we're predominantly having to deal with. You will see some variability certainly between different constructs and different serotypes, certainly. But when you're comparing a specific product, there we do see really strong consistency between batches.
Okay. And does Oxford Biomedica Solutions make use of HPLC and mass spec for AAV characterization?
Yes, we do. We do use both. We do a lot of mass spec to really understand and to see what's happening. We use both of those when we're doing a lot of force degradation studies, also to challenge to try to figure out what's likely to break down first. We also do a lot of analytical ultracentrifugation to look at the vector profile, empties, partials, fulls, and again that method we've qualified actually for GMP use to release product. Mass spec and HPLC predominantly we use for characterization, as you suggest.
Okay, well, I think that's about it for time. So thank you very much, Tim, and thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Bye, everyone.